Hello everyone, I'm Sarah and I did my project on cell regeneration. So, what is cell regeneration? Regeneration is the natural process of replacing or restoring damaged or missing cells, tissues, organs, and even entire body parts to full function in plants and animals. Scientists are studying regeneration for its potential use in medicine, such as treating a variety of injuries and diseases. Researchers also hope to learn more about the human aging process through studies of regeneration. This rapidly advancing field is called regenerative medicine. What is regenerative medicine? Scientists in regenerative medicine study the molecular, cellular, and developmental processes that control the development of new, healthy tissue. There are a few objectives of the field. The first one being to understand the molecular and cellular mechanisms of regeneration where it occurs naturally and how these mechanisms differ from the mechanisms of scarring. The second is to use the knowledge gained from this mechanistic understanding to develop therapies that will stimulate functional regeneration of damaged human tissues that do not possess the capacity to regenerate on their own. Now let's take a look at the history of regenerative medicine. Records dating back to the 8th century confirm the theory of regeneration was indeed acknowledged, although not entirely understood. Humans and animals have been utilizing these means since the dawn of creation, and now scientists in the field of regenerative medicine seek to move beyond the sole application of medical techniques and onto understanding the concepts, patterns, and mechanics behind regeneration to create more opportunities for comprehensive therapies that may one day be able to send an amputee home with a functional leg or relieve a diabetic from insulin injections by replacing the isolates within the pancreas. In 1992, futurist Leland Kaiser coined the term regenerative medicine. Regenerative medicine has grown from prior activities, including surgery, surgical implants, and increasingly sophisticated biomaterial scaffolds. Stem cells actually play a very important role in regenerative medicine. Stem cell research and therapies continue to enhance the field of regenerative medicine and what it can offer to patients and scientists. A portion of regenerative medicine research revolves around the use of stem cells, including embryonic, adult, and induced pluripotent stem cells. However, there are many other resources utilized in order to carry out the mission of regenerative medicine research. These include transplants, biomaterials, scaffolds, machines and electronics, stimulation pathways, and drug therapy. There are three main roles of stem cells that I want to elaborate on. The first one is that because of their role in development and their potential to develop into many different cell types, stem cells are vital to the field of developmental biology. Developmental biologists seek to uncover what genes and pathways are involved in cell differentiation and how these can be manipulated to create new healthy tissues. Secondly, stem cells can be applied to drug testing and development. New drugs that are developed could be safely and effectively tested using differentiated stem cells. As scientists learn more about how stem cells develop to form new tissue, they will be able to apply their knowledge in maintaining differentiated cell types that can be used to test particular drugs. And third, and of most interest to patients and scientists, is the role stem cells will play in cell-based therapy. These therapies will apply the understanding of stem cell development, differentiation, and maintenance to generate new, healthy tissue for diseases needing transplant or replacement of damaged tissue, such as arthritis, Parkinson's disease, type 1 diabetes, and coronary disease. Cell therapies may one day be able to replace organ donation and eliminate the issues that accompany it, such as rejection and tissue insufficiency. So, what kinds of organisms can use regeneration? The ability to regenerate is widespread in the animal kingdom, with representatives from most animal phyla displaying the ability to regrow missing body parts. All living organisms regenerate as part of the natural process to maintain tissues and organs, but some animals have extensive regenerative abilities. Tiny freshwater animals called hydra can form two whole bodies after being cut in half. More complex animals, such as mammals, have limited regenerative capacities. These include forming thick scars in tissues and skin to promote the healing of injured or amputated body parts, regrowing hair and skin, and knitting together fractured bones. I wanted to give examples of common organisms that use regeneration a lot. Some of the most common would be lizards, salamanders, axolotls, starfish, and sea cucumbers. Both lizards and salamanders can regrow a lost tail to full length. This process sees cells migrating to the wound, then slowly regenerating the tail within a few weeks. The finished appendage is completely functional and has all the features of the original, with the spinal cord and nerves growing back too. The axolotl is a relative of the salamander, but is even more skilled at regenerating. They can regenerate not just its tail, but also limbs, skin, and almost any other body part. Researchers found that each time a limb was removed, it regrew almost perfectly. This creature has since become the focus of research into human limb regeneration. The starfish can regrow arms that detach from their central disc, and some species have been observed to complete the process in reverse, regrowing an entire body from a lost arm. To do this, the starfish will store nutrients in the arm until it is able to regrow its mouth. Lastly, the sea cucumber has a remarkable ability to mend its organs in short periods of time, regrowing damaged parts and healing deep wounds in as little as a week. This is a vital skill for the sea cucumber. However, as in order to defend itself from crabs or turtles, the creature often jettisons certain organs as a distraction technique. My peer-reviewed article was on the mechanisms of regeneration. Regeneration involves the capacity for renewal or recompositing of tissues, organs, or even organisms after considerable physical injury or damage, resulting from pathologies, tumors, congenital diseases, or traumas. As a consequence of tissue regeneration, both the composition and the tissue properties are restored, and the newly formed tissue is highly similar to the original tissue. The regenerative capacity is directly related to the presence of stem cells or progenitor cells, which are capable of proliferation and differentiation. Tissues that maintain a high proliferative capacity, such as the hematopoietic system, have regenerative capacity even in adult organisms. I think it is important to explain what proliferation is. Cell proliferation is the process by which a cell grows and divides to produce two daughter cells. Cell proliferation leads to an exponential increase in cell number and is therefore a rapid mechanism of tissue growth. Cell proliferation occurs in repair processes in general, accompanied by intense production of extracellular matrix with large amounts of collagen, resulting in the formation of fibrous tissue to occupy the injured area. Tissues with greater regenerative capacity, such as the skin and liver, intrinsically present cells able to migrate to occupy the affected region, and the same cells maintain their proliferative capacity, enabling occupation of the lesion. This diagram shows the sources for new cells in regeneration. For the side labeled A, the top row shows that stem cells self-renew and produce one or more differentiated cells. The middle row shows de-differentiation, which is the process by which a cell loses differentiated character to produce a progenitor cell that can divide to produce more differentiated cells. The bottom row shows transdifferentiation, which involves the change of one cell type into others. This could occur without division or following de-differentiation of one cell type into progenitor for additional cell types. As a whole, the side labeled B shows distinct ways for accomplishing tissue-level pluripotency. The left side shows a pluripotent progenitor cell, which produces differentiated progeny cells spanning multiple germ layers. There could exist multiple and or self-renewal intermediates along different lineage paths. The right side shows different lineage-restricted progenitor cells, which each produce different differentiated cells. Each different tissue separately generates or harbors a restricted stem cell. These stem cells together can reconstitute the three different tissues, while any individual on its own is not sufficient. So, where can regeneration take place? Firstly, regeneration can take place on the molecular level. Cell turnover and division is regulated on molecular level. Inside all of your cells, there are hundreds, if not thousands, of small molecules interacting to drive the everyday processes that your cells must undergo to survive and function. In the context of regeneration, each cell's fate is dependent on these molecules and their interactions to drive them to divide, to remain the same, or to die. Cells adjust the rate at which these mechanisms are activated to control growth, replication, and death in response to their environment. Secondly, regeneration can take place on the cellular level. The cell is the body's most basic functional entity. Cells can either be homogeneous or heterogeneous, and almost always bundled together to create a tissue that has a specific structure and function. There are cells, for example, single nerve cells or neurons, that are capable of regeneration. Neurons consist of a cell body, an axon, and dendrites that work together to receive, process, and deliver messages all over the body. The axon has the ability to close the wound, generate a new growth place, and begin to regrow another extension to replace the damaged portion which is degraded. Lastly, regeneration can take place on the tissue level. Many tissues in the human body are capable of exhibiting regeneration in response to injury as well as general upkeep of normal tissue. To name a few, blood, skin, bone, skeletal muscle, liver, pancreas, small blood vessels, and kidney epithelium. In a perfect world, these tissues would be able to regenerate under any circumstance. However, the environment within the tissue must be just right. Cells that are able to generate energy, engage in crosstalk, and respond to the regeneration signals produced must be present. Additionally, the cells must be able to communicate in a stable and organized fashion, so they can be directed to proliferate and differentiate into the appropriate tissue. Finally, those signals that may inhibit regeneration must be modulated to enable an environment for growth and expansion of functional, healthy tissue. Now let's take a look at how cell regeneration takes place. The first way is called compensatory hyperplasia. In order for the organism to survive while a damaged liver regrows, the hepatocytes must be able to continuously divide while maintaining most or all their functions, including blood filtration, metabolism of toxins, and bile secretion. Scientists are currently trying to understand how and why hepatocytes are capable of this increase in cell production, while still maintaining or even increasing their function to compensate for the loss of tissue. The second way is by activation of resident through circulation adult stem cells. Adult stem cells are a subset of cells that are capable of self-renewal and can differentiate into many different cell types. These cell types are present throughout the organism's lifespan and have several features. The first feature is that they are self-renewing. Typically, when ASCs are triggered to divide, they undergo disproportionate division or asymmetric mitosis. One cell remains an adult stem cell, and the other goes on to become whatever cell is needed at that time. The second feature is that ASCs can be picky. Unlike embryonic stem cells, ASCs are not able to differentiate into just any type of tissue. The ability of an ASC to develop depends on its stage in life or the tissue that houses it. The third feature is that family is everything for ASCs. ASCs rely on their home, or what scientists call microniches, to maintain their stem cell capabilities. These microniches create an environment of free and open communication by acting as liaison between the stem cells and the rest of the tissue by sending and receiving soluble and insoluble signals. The niches are important for three reasons. One being that they keep ASCs happy and in a state of stemness, where the cells can maintain their status as a stem cell. The second being that they keep the ASC's feet on the ground. The connection between the ASCs and their microniche environment is essential for their function. When scientists mutate genes that disconnect ASCs from their niche, they can no longer remain stem cells. The third being that the microniche helps determine whether a stem cell will divide asymmetrically or symmetrically. The third way regeneration takes place is the creation of stem cells via de-differentiation. Cells that have already developed to their end fate are capable of turning back into adult stem cells. These cells give up their current occupation to return back to its position as stem cell. This is seen primarily in low-order vertebrates, such as fish, lizards, and amphibians. These organisms are able to regenerate limbs, tails, fins, jaws, parts of the eye, nerves, and intestines. The last way that regeneration could take place is signaling pathways inside and between cells. At any given time, millions of cells in our body are hard at work, maintaining our daily functions and helping us to do all the things we love to do. Cells work by using signaling pathways to talk to themselves and to other cells within the body. It is this communication that enables our system to operate as the giant symphony we know as the human body. For example, the endocrine system is spread over 8 different organs throughout the body that must communicate with each other on a constant basis. These organs use signaling pathways throughout the bloodstream to interact and function as an entire system. All stem cells use both internal and external transduction pathways in order to interact with each other as well as other cells in their microniches. These pathways will regulate division patterns of the stem cells. That's my works cited, and thank you for listening.